Hi, and thanks for watching this third segment of the lecture material on muscle. So our lecture material is presented in 3 recorded segments. This is segment number 3 of 3. And so far we have been talking mostly about muscle cells and the physiology of muscle contraction, the events that occur at the neuromuscular junction, and the physiology underlying the phenomenon known as muscle fatigue. So we've been thinking mostly about muscles at the cellular level. In this lecture, we're going to zoom out and think, yes, we're going to think about muscles at the cellular level, but then we're going to zoom out to think about muscles as organs and how these organs control body movements. In this segment, we will cover all of the topics shown here, starting with different types of muscle contraction. So in order to understand muscle contraction, here's a little initial vocabulary. Tension is the force exerted by a contracting muscle. And then load is what the contracting muscle, it exerts its force upon. I'll show you an image in the next slide, but tension and load are opposing forces, meaning in one sense, they really pull on each other. And in order for a muscle to move a load, the muscle must generate enough tension in order to overcome the load. So let's start with the images on the left side of this diagram. So we see a muscle that has been dissected out of an organism, and it's been connected via its tendons to a stationary point at the top. You can think about that as representing the muscle origin, right? The point of attachment to the less movable bone. And then down below, the other tendon is connecting that muscle to a load. In this case, it's a 3-kilogram weight. And the image all the way on the left shows the muscle length when the muscle is completely relaxed. So it's not generating any tension. That load is resting on the floor, so to speak. And that little light blue arrow that shows the events that occur as the muscle moves from the relaxed state to the contracting state. Notice that the load is rising. So in order for that muscle to move the 3-kilogram load, that muscle must have generated at least 3 kilograms of tension. And so now I'd like you to move over and take a look at the graph on the right. And so in the graph on the right, there's really 2 graphs here. The blue line represents the tension that the muscle develops over time, and the green line represents the length of the muscle over time. So let's start at time 0, where we see that bidirectional red arrow. At time 0, the muscle is developing no tension, and the muscle is at 100% of its resting length. And then a muscle stimulus occurs. And you remember what that is. That means an action potential was delivered to the muscle, and we'll talk a lot more about that today. But ultimately, stimulation arrived at the muscle, and that stimulation came in the form of action potentials, and those signals were transmitted to muscle cells. That's what's meant by muscle stimulus here. And so the muscle began to contract. And so when you see the blue line, that tension increases. But notice at the onset or just past the muscle stimulus that the green line remains flat for a little while. So at the onset of tension increases, but the muscle length does not decrease. So the muscle stays the same length even though the muscle cells are contracting. And then as tension continues to increase, so we're back to the blue line, and as we achieve a tension of at least 3.0001 kilograms, then that's enough tension to overcome the load. And during that time, the muscle will actually shorten. So now go back down to the green line, and we see that the muscle length is decreasing as the muscle is shortening, because the muscle is generating at least 3 kilograms of tension, and that weight is rising. So there are several different types of muscle contraction. So muscles generate tension, they pull on whatever they're attached to. And a muscle can generate tension while it's getting shorter, and we just looked at an example of that in the previous slide. A muscle can also generate tension while it's getting longer, and then a muscle can also generate tension while it is not changing length. So here's some new vocabulary. An isotonic concentric contraction refers to a muscle contraction where the muscle is generating the same tension over time and the muscle is getting shorter. So here, perhaps you'll recognize that that muscle that we're using as a model for basic understanding, that muscle is the biceps brachii. And I hope you remember that that muscle is attached to two different bones in your body. The origin of the biceps brachii occurs on the scapula, the shoulder blade, and the insertion occurs on the radius, one of the bones of the forearm. And when that muscle contracts, it causes a body movement called flexion of the forearm. So the elbow will bend, right, as shown by the direction of that arrow. So why is this isotonic concentric contraction? Iso means same. So while this person is lifting the weight, let's make up that that's a 10-pound weight. This muscle must be generating at least 10.0001 pounds of tension. And so that muscle, in order to move that weight, that load, it's a constant load, that muscle must be generating a constant tension, 10.0001. And so that's why we call it isotonic, same tension. And then concentric means that the muscle is getting shorter. And as that muscle gets shorter, the insertion is pulled toward the origin. The radius is pulled toward the scapula, and that's why the arm bends at the elbow. That's isotonic concentric contraction. What happens next? You just finished that isotonic concentric contraction. You've just completed a bicep curl, and what do you do next? You unbend your arm at the elbow. That's called extension of the arm at the elbow. And this movement is called isotonic eccentric contraction. In order to move the radius further away from the scapula, that muscle, the bicep brachii, needs to get longer. And that muscle is still generating tension. So isotonic means same tension. We're still holding a 10-pound weight. And in this case, the muscle is getting longer. That's eccentric contraction. And it's important to note that the muscle must be generating tension here in order to slowly lower the weight. That's the safer movement. Consider slowly lowering the weight using isotonic eccentric contraction. Consider doing that versus at the end of a bicep curl, the bicep muscle relaxes completely. There's a risk of injury there, right? As your arm extends in an uncontrolled fashion, you could injure the elbow joint. So this is an example of muscle lengthening while it is generating a constant amount of tension. And then it's also possible for muscles to generate tension, but to not change length. So now this person is just holding the weight there. There's a little bit of flexion in their elbow. The elbow is slightly bent, but their arm is fixed and stable. They're holding the weight at the same position. And so the muscle is generating tension and the muscle is remaining the same length. And right now, the weight of that barbell, that hand weight, is remaining constant, right? We decided earlier that it was 10 pounds. And so the tension is not fluctuating here, but let me give you an example of a condition where the tension would fluctuate and the muscle would be staying the same length. I'll have that example for you in a moment. First, let's reinforce some vocabulary. This person is doing bicep curls using a hand weight. So when she raises the weight, as shown in the upper arrow, what name would we apply to that body movement, to that muscle movement? And then when she lowers the weight, the bottom arrow, she's extending her arm at the elbow. She's not just dropping the weight. She's doing it in a very controlled fashion. What would we call that muscle movement? And how about this yoga pose? Trikonasana or triangle pose. This is a very difficult yoga pose. You can get really winded by holding this pose, by staying perfectly still for, say, a minute or two. This is a difficult pose. And this is another example of isometric contraction. The muscles, while she is holding this pose, the muscles are keeping the same degree of tension, and the muscles are also staying the same length. We can use this image of a food server to consider what happens during isometric contraction. So the muscles remain the same length, but the tension that the muscles are generating will change over time. So think about this tray full of, I don't know, those look like perhaps espressos. So the food server goes over to the table and puts the espressos down on the table while maintaining the position of the tray. Consider what's happening to the length of the biceps brachii as well as the tension that the biceps brachii is generating. So the length of the biceps brachii, what happens? Nothing. The length of that biceps brachii remains the same because the food server is holding that tray in a constant position. So the joint angle is not changing. The muscle is not getting longer. The muscle is not getting shorter. But as that food server puts the cups of espresso down on the table, what happens to the amount of tension that the biceps brachii is generating? It goes down, right? As the tray gets lighter, less tension is required to overcome a lightening or a lessening load. So this is an example of isometric contraction in which the tension generated by the muscle varies according to changes in the load. Here's another example of that same phenomenon. So somebody is holding up their water glass and a different person is pouring water into that water glass. In order for this whole process to work, the water glass needs to stay still. So what's happening to the length of the muscles that are responsible for holding that water glass still? Nothing. Muscle length is not changing because the position of all those joints is stable while water is being poured into the glass. What is happening to the tension that those muscles are generating over time? It's increasing. The muscles that are being used to hold up that glass and to keep that glass still, those muscles are facing an increasing load as more and more water comes to fill the glass. And so in order to keep that glass still, those muscles must generate matching tension. So this is an example of isometric contraction where the tension is variable, and here the tension that the muscles are generating is increasing over time as the load increases. And here we can summarize those three new vocabulary terms. So in panel A, we see concentric contraction. So the muscle is shortening while maintaining a constant tension because the weight of the book, the load, remains stable, but the muscle is getting shorter. In panel B, we see eccentric contraction. The muscle is getting longer and the load is remaining constant. And then in panel C, we see isometric contraction and the load is remaining stable. The muscle length is remaining stable as that person just holds the book out steady. This is a fantastic, cute video to reinforce these different types of muscle movements. She does a great job of explaining these concepts, and I'll encourage you to pause this video, go into the tutorial resources in Canvas, and this link is right there near the top of the video tutorials. It's definitely worth 2.5 minutes of your time. We'll spend the next several minutes talking about the factors that influence how much tension a muscle can develop and how much tension a muscle will actually develop. And so in order to understand total muscle tension, we need to begin at the cellular level. So under number 1 here, we'll talk about some factors that influence how much tension an individual muscle cell, remember a muscle cell is also called a muscle fiber, how much tension an individual muscle cell will generate. And there are several factors that influence that. And then we'll move on to heading number 2 here, the number of muscle cells that are contracting at any time and what influences that number. So in order to dive into point number 1 here, the amount of tension developed by each fiber, we need to revisit what's happening at the level of an individual cell. And when we look at muscle contraction in one individual cell, it's called a twitch contraction. A twitch contraction is the mechanical response of a single muscle cell following a single muscle action potential. These are the phases of a twitch contraction. So on the vertical axis here, we see force of contraction. It's perfectly fine to think of that as tension. How much tension is the muscle generating? And then on the horizontal axis is time. A twitch contraction begins at here, it's time equals 0, where we see the black arrow, and the black arrow indicates the onset of a muscle action potential. So that means an action potential is traveling along the plasma membrane of a muscle cell and down the T tubules. After that, we see the latent period, and we've covered the latent period before. It includes, or the explanation for the latent period is, events need to occur inside the muscle cell before we actually observe any muscle contraction. So I hope you remember that during the latent period, the mechanisms of excitation, contraction, coupling occur. And then we move into the next phase, which is the contraction period, shown here in red. And during the contraction period, muscle tension increases. And following the contraction period, there's the relaxation period. During the relaxation period, muscle tension decreases and eventually goes back to the resting tension. These are the 3 phases of a twitch contraction. A twitch contraction begins with the latent period where there is no increase in muscle tension. It lasts for about 2 milliseconds, and that's the time required for calcium to be released from the sarcoplasmic reticulum. Calcium binds to troponin. The contraction cycle begins. And so that is the mechanism of excitation-contraction coupling. And after the contraction cycle gets underway, we move into the second phase, which is called the contraction period or contraction time. And this is when the tension that the muscle cell develops increases over time. And then the contraction time is followed by the relaxation period. And during the relaxation period, the muscle action potential is over with. The calcium release channels in the sarcoplasmic reticulum close. Calcium pumps pump the calcium out of the cytosol and back into the sarcoplasmic reticulum. And the troponin-tropomyosin complex comes to, once again, block the myosin binding sites on actin. The sarcomeres get longer as the thin and thick filaments slide past each other in the opposite direction. That's the relaxation period. So in order to understand the total amount of tension that a muscle will develop, we need to understand a little bit more about twitch contractions. So we're still talking about muscle contraction in one muscle cell. And one thing that can influence the degree of contraction, the intensity of contraction in a single muscle cell is the frequency of stimulation. So far in a twitch contraction, I've told you that it is the muscle contraction that results from a single muscle action potential. Well, what if that muscle cell is stimulated sequentially? What if a second action potential occurs before the muscle fiber is fully relaxed? That will lead to an even stronger tension developing in the muscle cell, and that process is called wave summation. So we're looking at a similar graph here. Let's check out our axes. On the vertical axis is force of contraction or muscle tension. It's totally fine to think about it either way. And on the horizontal axis, we have time. Even though it's not labeled, we know that that's time. And so far, we have looked at the events that occur during a twitch contraction, and that's panel A on the left. And here we see not only what happens during the twitch contraction, but we see the electrical events that occur in the muscle cell. So instead of indicating the onset of a muscle action potential using a black arrow, we can actually see the graph of the muscle action potential. So in a twitch contraction, that muscle action potential is followed by the latent period, the contraction period, and the relaxation period. And that's a twitch contraction. Let's move over to the wave summation, where now we have arising two muscle action potentials in sequence. And so the first part of the wave summation graph, where we have our first muscle action potential, the force of contraction rises as it would ordinarily under a twitch contraction, and it begins to fall. Only during the relaxation period, a second muscle action potential occurs. And this second muscle action potential occurs before that muscle cell has fully relaxed. So some of those cross bridges have still formed, and this will lead to the release of additional calcium from the sarcoplasmic reticulum. And notice that following that second muscle action potential, a much greater force of contraction is observed in that muscle cell. We can add on to that. So what we were just looking at is panel A and panel B on the left. What happens if those muscle action potentials keep on coming? So now we'll look at panel C. It's a condition known as unfused tetanus. So it's very similar to panel B, where we see the first two muscle action potentials, but what if we get two more muscle action potentials? And at each point, these muscle action potentials occur before the muscle cell has had the time to fully relax. Notice that this sequence of action potentials leads to ever increasing force of contraction. Each successive action potential causes the muscle cell to generate more and more tension. That's unfused tetanus. And we call it unfused because we can observe in the force of contraction that relaxation begins. The muscle cell begins to get a little bit longer before the contraction, the force of contraction increases again. So we have this sort of humped-like shape to the graph. Let's move over to panel D, which is unfused tetanus. Look at the frequency with which the muscle action potentials occur there. They occur much more frequently. So there's a shorter space of time from one action potential to the next. And what that leads to is the pattern of contraction that we see in the red line. So the muscle will begin to contract, and before it's even started to relax, that muscle cell undergoes another muscle action potential. And that immediately causes a further increase in the force of contraction. So in between these action potentials, in fused tetanus, we see no signs of the muscle cell relaxing. We only see that the force of contraction increases over time with each successive delivery of a muscle action potential. Also, in fused tetanus, we see that plateau at the top, the flat part of the red line. So that plateau represents maximal tension or maximal force of contraction. We can continue to deliver repeated muscle action potentials, but the force of contraction has already hit its maximum. It doesn't go any higher. And then, of course, relaxation will occur when muscle action potentials cease. So to review, the frequency of muscle stimulation, the frequency of those muscle action potentials can influence the overall force of tension or the overall force of muscle contraction, the amount of tension that results. Unfused or incomplete tetanus occurs when a muscle fiber is stimulated between 20 and 30 times per second, and the muscle cell has the opportunity to only partially relax before the next muscle action potential occurs. And so, as we saw on the graph before, this results in sustained muscle contraction, but it's wavering. The tension that gets developed in that muscle cell increases, and then it decreases a little bit, and then it increases more, and then it decreases a little bit, and then it increases even more. So that's sustained tension, but it's wavering. It goes up, and then it comes down a little, and then it goes up, and then it comes down a little. And in contrast, we can also look at fused or complete tetanus. And tetanus occurs when a muscle fiber is delivered at a much when a muscle fiber is stimulated at a much faster rate, so 80 to 100 times per second. The muscle cell does not have an opportunity to relax at all before the next muscle action potential occurs. And so this results in that sustained contraction, and it's not wavering. And it's called fused tetanus because the individual twitch contractions, the contraction followed by a brief window of the onset of the relaxation period, that doesn't occur. We never observe that brief window of relaxation. So rather, we observe this sustained contraction with no evidence of relaxation. And so those muscle contractions, those twitch contractions, one way to think about it is they have now been fused together on the graph such that we no longer see any evidence of muscle relaxation. Another factor that influences the amount of tension that a muscle will develop, and we're still talking about the cellular level here, is, well, how long is that muscle when the muscle action potential occurs? And this is known as the length-tension relationship. And we can think about it in terms of, just looking at a single muscle cell, just looking at a single sarcomere. And then we can zoom out and think about how that might look at the level of an entire muscle, like the biceps brachii. So let's examine the length-tension relationship. I hope you remember that sarcomeres get shorter and they get longer. That a sarcomere is a portion of a myofibril, and it is bounded by a Z disc on either side. So during contraction, whether it's partial contraction or maximal contraction, the sarcomeres get shorter. And if we look at the level of the muscle, in this case, the entire biceps brachii, as those sarcomeres get shorter, the insertion of that muscle, the radius, is going to move toward the origin, the scapula. We can also think about sarcomeres getting longer. So sarcomeres will get longer when a muscle is stretched, when the origin is pulled away from the insertion. And so the distance from one Z disc to the next Z disc will actually increase. And so we know that these sarcomeres vary with muscle position. If the muscle is shorter, the sarcomeres are shorter. If the muscle is longer, then the sarcomeres are longer. So it turns out that the length of a sarcomere at the beginning of a contraction influences the degree to which that sarcomere can shorten or can generate tension. It'll take a little while to understand this concept. So we're looking at a graph of what's known as the length-tension relationship. So on the vertical axis, we see tension developed, and that's expressed as % of maximum. So when there's zero tension, the muscle is absolutely not contracting, and when there's 60% tension, that means that the muscle is contracting at 60% of its maximum. And then let's look at the horizontal axis. The horizontal axis is resting sarcomere length, % of optimum. And we're going to talk about what optimum means in just a moment. But for now, the horizontal axis is resting sarcomere length. So is the sarcomere short? Is the sarcomere long? Is the sarcomere somewhere at a medium length when muscle contraction begins? So think about this as, I don't know, look at your bicep brachii right now. For one of your arms, how long is your bicep brachii? Just sitting in the position that you're naturally in right now. Is your bicep brachii particularly long? It would mean that your your arms are straight, your elbow is not bent at all. And for most of us, we're not going to be sitting here watching a lecture video with both of our arms extended at the elbow. It's just not a natural body position for most of us. So I'm going to guess that the sarcomeres in your bicep brachii right now are on the shorter side, probably because your arms are bent right now. And so that's what I mean. What is the sarcomere length at the beginning of muscle contraction? And down below that, below the vertical axis, we see some labels: understretched, optimal length, overstretched. So as our sarcomeres are shorter, we call that understretched, and that is a suboptimal length for generating maximal contraction. And then above the optimal length, that means our sarcomeres start out in a stretched position, and that is also not optimal for generating maximal muscle contraction. And then in the middle there, we have the optimal length. The optimal length of a sarcomere is the length at which maximal muscle contraction is possible. So notice that for understretched and overstretched, we never achieve 100% of maximum tension, but we do achieve 100% of maximal tension when the sarcomere is at the optimal length. So let's actually look at some sarcomeres here, and let's look at what's happening in this understretched sarcomere. So this is the length of the sarcomere at the onset or the beginning of muscle contraction. So this sarcomere is clearly already shortened, right? There is no H zone. The thin filaments are overlapping each other. There is no H zone. So this muscle, this sarcomere, is partially shortened already. And then a muscle action potential occurs. And then the contraction cycle begins. Because this sarcomere was understretched at the onset of those events, at the beginning of the muscle action potential, it means that there's less room for the sarcomere to further shorten. And let's think about that. Do we see these thin filaments here and their relationship to each other, as well as the relationship to the thick filament? There's less opportunity for those thin filaments to be grabbed by the thick filament. The myosin heads have less opportunity to grab onto actin. And because the sarcomere is already in a shorter position, there's less room for that accordion-like movement for this sarcomere to get even shorter. And we'll contrast that with this sarcomere. Let me change colors. With this sarcomere here, that's starting off in the optimal length. So what's going on here? At the onset of a muscle action potential and at the beginning of the contraction cycle, notice the spatial relationship here between the thick and thin filaments. There's lots of opportunity for cross bridges to form, for those myosin heads to bind to actin. And then there's also lots of opportunity for those filaments to slide past each other. So there's lots of opportunity for the physics, the pulling action, and there's lots of opportunity for the shrinking, the narrowing of the width of the sarcomere. And so these spatial positions allow this muscle cell to achieve 100% of the tension possible. So let me change colors again, and let's think about what happens in this overstretched sarcomere. So this muscle is already being stretched, the sarcomere is longer than optimal, and then the muscle action potential occurs, the contraction cycle begins, and how come we're not able to achieve 100% of maximum? Well, there's lots of room to move laterally, right? There's plenty of opportunity for the sarcomere to shorten, but look at the spatial relationship between thick filaments and thin filaments. There's not a lot of opportunity for the myosin to grab hold of the actin. Very few myosin heads have the opportunity to form cross bridges. These myosin heads here and these myosin heads here, because the H zone is so wide, those myosin heads have nothing to grab onto and cannot play a role in muscle contraction. We can do a little demonstration using your own body to understand the length-tension relationship. So just take your hand and extend it, and then make a fist. Make a fist, make the tightest fist you can. Feel the muscles, use your other arm to grab your forearm, and feel those forearm muscles contract as you're making a fist, right? The muscles of your forearm control the bones of your fingers. So as you flex your fingers into a fist, what's causing that movement are the muscles of your forearm. So we can change the length of those forearm muscles and then have you make a fist, and you can experience the length tension relationship. So extend your arm at the elbow, straighten out your arm at the elbow, and keep your hand in the neutral position. So put your hand in position B, and now make the tightest fist you can possibly make. And feel that. Or maybe your knuckles are getting lighter in color. Maybe you can feel the muscles in your forearm bulging as you make this really tight fist. Okay, so that is when your hand is in, your wrist is in the neutral position. Next, take your hand and put it in position A. Think about what's happening, and before you make a fist, think about what's happening to the length of the muscles in your forearm. Those flexors in your forearm are getting shorter, right? Just by you flexing your hand at the wrist into position A, those muscles are getting shorter. And now, with your hand in position A, make a fist. Make the hardest fist you can make. It's not as strong as the fist was when your hand started off in position B. So we're starting in the understretched position. And now let's go over to position C. So put your hand into position C and now make a fist. It too is also, those muscles are not able to generate as much tension because those muscles in your forearm start off in an overstretched position. The sarcomeres are extra long, and because they're so long, those muscle cells are not able to generate enough tension, or they're not able to generate maximal tension. So in position B, that's the neutral position, those sarcomeres are at their optimal length and can generate 100% maximal tension. But in position A, the sarcomeres are too short, they're understretched, and in position C, those sarcomeres are too long, they're overstretched. This is the length-tension relationship, and it's important in skeletal muscle, and it will come up again for us when we talk about cardiac muscle down the road. Another factor that can influence the amount of tension that a muscle cell generates is, well, how big is the muscle cell? And I don't mean how long, how wide. The diameter of the muscle fiber. It turns out that fatter muscle fibers, larger diameter muscle fibers, can generate more force because they contain more myofibrils. So they have more ligament filaments, they have more myosin heads that can pull on more actin, and in that way they can generate more force. Here we see an image published as part of a scientific report titled Influence of Feeding Regime and Finishing System on Lamb Muscle Fiber and Meat Quality. And the content of this paper is not so important for us, but I'm showing it here as an example of 3 different muscle fiber types. And most importantly for us right now, we can see that muscle fibers differ in diameter. How wide are they? So we will continue on this discussion to talk about the differences between the FG fiber types, the FOGs, and the SOs, but I do want to point out that there are differences in diameter. So how about, let's use green here. And so here's an FG muscle fiber, except that's purple. That's okay. Here's an FG muscle fiber, and here's an FOG muscle fiber, and notice that they have 2 different diameters. This FG muscle fiber is wider in diameter because it has more myofibrils than this FOG muscle fiber. We'll talk more about fiber diameter in just a little bit, But let's take a moment to review the 3 ideas that we have talked about so far, and then we'll add in the degree of fatigue, which we have talked about recently. So we are interested in the total muscle tension that a muscle develops, and that is influenced by factors that affect individual cells, and that's what we've been immersed in for the last few minutes, and it's also affected by factors that happen at the level of the muscle, and that's where we're going to go next. So thinking about the cellular level, each muscle fiber, right, is each muscle cell. We can influence total muscle tension by modifying the frequency of muscle action potentials. How often is that muscle cell stimulated? We can influence total muscle tension by adjusting the length of the sarcomeres at the onset of the muscle action potential, and that's called the length-tension relationship. Another factor that influences total muscle tension is the diameter of the muscle fiber, and we'll talk more about the different fiber types in just a moment, but the larger the diameter, the more tension that type of cell is able to generate. And then the last thing that we can add on here is the degree of muscle fatigue. And we talked about the biochemistry of muscle fatigue earlier. It's not thoroughly understood, but we have some good ideas about why muscle fatigue happens. And I'll remind you that some of the ideas around muscle fatigue include that the ingredients required for excitation contraction coupling become depleted. So the central nervous system continues to stimulate the muscle or the muscle cell, except the motor neuron might become slightly depleted in acetylcholine. The sarcoplasmic reticulum might become slightly depleted in calcium, and other factors as well. Next, we're going to move on to talk about the number of fibers contracting in a muscle at any given time. So if you go and pick a pen up off the desk, how many muscle cells in your bicep brachii are contracting in order to lift that pen versus you want to lift a 30-pound weight using your biceps brachii, how many muscle fibers are contracting in order to lift that 30-pound weight? So this brings us to the concept of the motor unit. A motor unit consists of one motor neuron plus all of the skeletal muscle fibers that one motor neuron stimulates. And motor units differ in size. So a small motor unit consists of one motor neuron and just a handful of muscle cells, maybe 10 or 20 muscle cells. And then we also have large motor units and everything between. A large motor unit might consist of one motor neuron plus thousands of muscle cells that contract in response to stimulation from that one motor neuron. So we can use this image to understand the concept of a motor unit. So up top we see a segment of the spinal cord and we see 2 different motor neurons. So motor neurons deliver efferent signals from the central nervous system to the effector, and in this case the effector is skeletal muscle. So we see the blue motor neuron A, and we see the green motor neuron B. And for each motor neuron, A and B, we can see that they innervate a different collection of muscle cells, all within the same muscle. So we can pretend that that muscle is biceps brachii, and we're looking at 2 motor units that are part of the biceps brachii. Let's look at motor neuron A, the blue motor neuron. Can you visualize the 1, 2, 3, maybe 4 muscle cells that are part of motor unit A? And then let's go and look at motor neuron B. Can you visualize the 1, 2, 3, 4, 5, 6, maybe 7 or 8? It's a little difficult to tell. Maybe motor neuron B forms a neuromuscular junction with 8 different muscle cells. And so motor unit B is larger than motor unit A. So motor units are operated independently. In that previous slide, it would be possible for the central nervous system to send impulses only along the blue neuron, motor neuron A, and not along the green motor neuron B. So motor unit A, those muscle cells would contract, while motor unit B, those muscle cells would not contract. And so when we activate an increasing number of motor units, we call that motor unit recruitment. We'll spend a bit of time using this image. And first, I'll continue to explain just the concept of motor unit recruitment using panel A. And then we can compare muscles that have different sized motor units by comparing panel A with panel B. So let's spend a moment focusing on panel A, where we're just thinking about any given muscle and the tension that that muscle generates based on the number of motor units that have been recruited, where recruited also means activated. So let's start off, maybe we'll pick dark blue. And on the horizontal axis here, we have the relative strength of whole muscle contraction. So is the muscle contracting weakly, or is the muscle contracting at its maximum tension? And so let's start with activating motor unit number 1. When we activate just one motor unit in this muscle, we get a step up in the total tension, right? Because this certain number of muscle cells begin to contract, and as they all contract and the other muscle cells remain relaxed, as this one motor unit is contracting, we get this level of tension that can develop. And let's say we need more tension. This muscle needs to contract harder, in a sense, in order to accomplish our goals. So we'll recruit motor unit number 2, and there is another stepwise increase in the total muscle tension generated. And if we need more tension to overcome a load, we can recruit motor unit number 3, and we can recruit motor unit number 4. All right, so with each motor unit recruitment, we get this stepwise increase in tension that develops. Now I want to compare panel A with panel B. And in panel A, we're looking at a muscle that has small motor units. And by small motor units, I mean each motor unit only includes a handful of muscle cells, maybe 10 muscle cells. And in panel B, we're looking at a muscle that contains very large motor units. So a large motor unit might include a few thousand muscle cells. And so let's pick some real muscles to compare. Panel A represents, or a good example of a muscle in panel A would be any of the muscles, any of your extrinsic eye muscles. So those are the muscles that are attached to the exterior surface of your eyeball. And they are also attached to the bones that make up your skull. And when those muscles contract, they pull your eyeball in the orbit. They pull your eyeball in different directions. So you can keep your head still and you can just move your eyes. You can look up, you can look down, left, right, etc. And we have exquisite control over our eye movements. And these small muscles have small motor units. And that's the example that we're going to use for panel A. Let's contrast panel A with panel B. And an example muscle here might be the rectus femoris. That's one of your quadriceps, one of the large muscles on the front of your thigh. And these muscles are very powerful. They have large motor units where one motor neuron might control 2,000 or 3,000 muscle cells. So let's look at panel B for a moment and look at the stepwise increase in the strength of contraction with motor unit recruitment. So here in the quadriceps, we recruit motor unit number 1, and look, we get this sort of giant increase in tension. We go from no tension to this amount of tension. And let's say we need to generate even more force. We recruit motor unit 2, and now maybe 2,000 more muscle cells begin to contract, and we get this relatively large step up in muscle tension. Let's say we need more force. We can recruit motor unit number 3. And again, another 2,000 muscle cells will become activated during motor unit recruitment. And so we can compare the increase in the relative strength of muscle contraction in a motor neuron, in a muscle that has, this is a muscle that has small motor units. So we get these tiny increases in force with every new motor unit that gets recruited. And we can contrast that with a muscle that has large motor units where we get these giant increases in tension that develop with every new motor unit that's brought online. And think about the difference in the degree of control that is exerted by your extrinsic eye muscles. How fine-tuned is the movement of your eyeballs? It's very fine-tuned. It allows you to read the words on a page. Versus how finely tuned is the movement of your rectus femoris, the muscles, one of the muscles on the front surface of your thigh. That basically extends your leg at the knee, and that's kind of all it does. It kicks your leg out, and that's it. So it's a much larger, more forceful, less refined movement. All right, so we're rounding out this fairly large topic. We're talking about the total amount of tension that a muscle can develop, and it's determined by factors that occur at the cellular level, and then it's determined by factors that occur at the level of the whole muscle. So how large are those motor units, and how many motor units have been recruited? How many of them are active? So this brings us back to talking about different types of muscle fibers, different fiber types. And we're going to talk about 3 different muscle fiber types, and they are classified according to these 2 criteria. So for any given muscle fiber cell, you can ask, well, is it fast or slow? And is it glycolytic or oxidative? What do those terms mean? Fast or slow refers to the type of myosin that occurs in the thick filaments of the cell. So there are 2 types of myosin in skeletal muscle cells, and they're named reasonably. Fast myosin has higher enzyme activity. So I hope you remember that the myosin head is also an ATPase. And so fast myosin is faster to cleave ATP. And you remember the cleaving of ATP re-energizes that myosin head and gets it ready for the next power stroke. So fast myosin can perform the contraction cycle about 4 times faster than slow myosin. Slow myosin, the myosin head is also an ATPase. It also hydrolyzes ATP to re-energize the myosin head, but that process happens more slowly. So the power strokes, the series of power strokes, happens more slowly in slow myosin. And then the second criterion we use to name muscle fiber types is glycolytic or oxidative. And so this refers to, well, what is the primary mechanism that this cell uses to generate ATP? And so glycolytic fibers rely mainly on glycolysis. They will have a lot of glycogen, and they won't have a lot of myoglobin, and they won't have as many mitochondria relative to oxidative fibers. Oxidative fibers produce ATP mostly through oxidative phosphorylation. So they will have a lot of myoglobin because myoglobin temporarily stores oxygen, and they will have more mitochondria because oxidative phosphorylation occurs inside mitochondria. So again, glycolytic fibers rely mainly on glycolysis, and so they have the ingredients present in their cytosol to perform lots and lots of glycolysis. And glycolysis takes many different enzymes, and those enzymes are upregulated. We find more of those enzymes in the cytosol of a glycolytic fiber than we do in an oxidative fiber. And because glycolytic fibers don't rely so much on oxidative phosphorylation, they have fewer mitochondria and they have less myoglobin, They are less well vascularized. Remember, it's the capillaries, the blood capillaries that allow oxygen to be delivered to our muscle cells. Well, glycolytic fibers don't have quite as high a demand for oxygen because they rely mostly on glycolysis, which is an anaerobic process. And because the glycolytic fibers have relatively little myoglobin and they're not as well vascularized, this muscle tissue can take on a light appearance. And so if you're a meat eater, you might call this white meat. And so a classic example of white meat is a chicken breast. Think about chicken breast versus chicken thigh. Chicken breast is the white meat and chicken thigh is the dark meat. And we can contrast glycolytic fibers with oxidative fibers. Oxidative fibers, because they rely on oxidative phosphorylation, they have a lot of the necessary tools to perform oxidative phosphorylation. Do they also do glycolysis? Yes, they do. But they do more oxidative phosphorylation than glycolytic fibers do. So in oxidative fibers, we're going to see more blood vessels. We'll see more mitochondria. There will be more myoglobin in the cytosol. And this myoglobin, as well as the surrounding blood vessels, give this meat a darker, redder appearance. And so this is dark meat. And this would be chicken thigh. Even before I talk about the information that's on this slide, I want to have you think about chicken meat and chickens for a little while. So we're thinking about chicken breast, the light meat, versus chicken thigh, the dark meat. And now think about the animal, a chicken. Do chickens fly? Do they spend a lot of time flying? No, they don't. They can fly a little bit to escape danger. They can maybe fly up onto the roof, but they're not migratory birds. They're not flying hundreds or thousands of miles every year. They're not really flyers. But what do they do all day? They walk around. So light versus dark has something to do with the fatigability of the muscles. How much endurance does a particular type of muscle cell have, or a particular muscle? And so which one is going to need greater endurance, the chicken breast or the chicken leg? The chicken leg. Chickens walk around all day long. They almost never stop walking around. They'll go and sit down at night, but for most of the day, they're walking around, they're feeding, they're having social interactions, and they do very little flying. So for most of the day, those breast muscles are not very busy. They're relaxed, and the leg muscles are almost nonstop active all day long. Okay, now we can talk about the information on this slide. So combining those criteria that we just talked about, fast myosin or slow myosin, and glycolytic or oxidative, we have 3 different muscle fiber types. Type number 1 are commonly referred to as slow oxidative fibers. In some textbooks and some research papers, they are actually referred to as type 1 muscle fibers. I'll always use the term slow oxidative, because I think it's just less for you to memorize. As a student, you can actually think about, well, what does slow mean? What does oxidative mean? Versus the term type 1 that has no intrinsic meaning. So slow oxidative fibers have slow myosin. So they take longer to reach peak maximal muscle contraction, and they rely heavily on oxidative phosphorylation. So they have lots of myoglobin, they're well vascularized, lots of mitochondria, and they are darker in color. The slow oxidative fibers tend to be small in diameter. So they're not particularly strong, they're not the most powerful types of muscle cell, but they do fatigue really slowly. They can perform aerobic cellular respiration all day long. Those mitochondria just keep cranking out the ATP, and they can go for hours and hours and hours without showing any sign of fatigue. Those are our slow oxidative fibers. And from number 1, I'm going to jump down to number 3 on this slide. And I'm skipping number 2 for a moment. Let's look at the opposite, number 3. Those are the fast glycolytic fibers. So these fast glycolytic fibers have fast myosin. They can reach peak tension in a shorter amount of time. And these muscle cells rely on glycolysis as their primary means for generating ATP. Glycolysis is faster, but it doesn't generate nearly as much ATP as oxidative phosphorylation. So these muscle cells don't have a lot of myosin. I'm sorry, they have plenty of myosin. These muscle cells don't have a lot of myoglobin. And so that makes them lighter in color. They're also not, the muscle is also not particularly well vascularized. So fewer blood vessels, less myoglobin. These muscle cells and these muscles are lighter in color, and they tend to be widest in diameter. They're big, they're fat, they're powerful. They are much stronger than your slow oxidative fibers. So fast glycolytic fibers are the strongest fibers in your body, but they also fatigue quickly. They do glycolysis. They do a little bit of oxidative phosphorylation, but they're not equipped to do a whole lot, so they'll run out of steam quickly. They'll be unable to replenish their ATP supply, so they'll get tired very quickly. And then we have the intermediate fibers, the fast oxidative glycolytic muscle fibers, and they're called intermediate fibers for good reason. They show some of the characteristics of the slow oxidative fibers and other characteristics from the fast glycolytic fibers. So they're sort of a medium fiber type. They're fast, so they have fast myosin, and they rely on a combination of glycolysis and oxidative phosphorylation. So they have moderate amounts of the ingredients needed for both types of cellular respiration, for both types of ATP production. So they tend to be medium in color or pink in color. They're not white, they're not red, they're pink because they have a moderate amount of vascularization and they have a moderate amount of myoglobin. And it turns out they also fatigue at a medium rate. So they have more endurance than the fast glycolytic fibers, but less endurance than the slow oxidative fibers. And they're also medium in diameter. So they are medium in the maximal amount of tension that they can generate. So here's another image through skeletal muscle tissue. These are muscle cells in transverse or cross section. And in the panel on the right, you can see the different fiber types labeled there. So the SO stands for slow oxidative, FG stands for fast glycolytic, and FOG stands for fast oxidative glycolytic. And we can see that there are clear differences in the diameter of each fiber type. The SOs are the smallest diameter, they are the weakest, but they have the greatest endurance. The FGs are the largest in diameter, they are the strongest, but they have the lowest endurance. They fatigue more quickly than the other two. And then the intermediate fiber types are the FOGs. So they're sort of medium in size and they're medium in their strength and their endurance. So in a muscle, like your biceps brachii, we're going to find many different motor units. For any motor unit, all the muscle cells in that motor unit are all the same fiber type. They're all FGs, they're all FOGs, or they're all SO fiber types. And so in a muscle, like the biceps brachii, there is a mixture of muscle fiber types, and the capacities of a given muscle, like the biceps brachii versus in your arm versus the rectus femoris in your leg, the properties of those two muscles are going to be determined by the ratios of different muscle fiber types. This is going to make differences in muscles in the speed with which they contract, fast fibers versus slow fibers, the strength, the force that they can generate, and how much endurance they have. How quickly do these muscles fatigue? In any particular skeletal muscle, there will be a ratio of different muscle fiber types. In that particular skeletal muscle, there will be a particular proportion of muscle cells that are SO fibers. Another proportion of the muscle cells will be FG fibers, and then the third proportion of muscle cells will be FOG fibers. And the ratios of those three fiber types will differ from one given muscle to the next, depending on how that muscle contributes to posture, body movement. So let's think about these three types of movements. Number one, we have muscles that are involved in maintaining your posture. So think about the muscles that hold your head up. You've been holding your head up since you got out of bed this morning, and you will hold your head up until you lay down next. Number two, let's think about muscles that work all day long without stopping. They work 24 hours a day, like your diaphragm. You never stop breathing. In order to move air into and out of your respiratory system, your diaphragm needs to continuously contract and relax, contract and relax. And then maybe you've been walking around all day. I hope you've been walking around all day. I find that I do a lot of sitting in the time of COVID. And then number three, you have muscles that are responsible mostly for fast, powerful actions. So such as lifting up a child, or throwing something, or jumping, or kicking. These are activities that we do quickly, and we do them once or twice, and then we stop doing them. These are not activities that we need to do all day long. So let's think about each of these example movements: postural joint stabilization, long-lasting repetitive activities, and fast, powerful actions. Let's consider those three categories of movements and overlay on top of that the relative proportions of the three different muscle fiber types. So some of these body movements require lots of endurance, and they don't require particular strength. So let's think about the muscles that are holding up your head and the muscles that are keeping you erect all day long until you lay down next. So the muscles of your back, like your erector spinae, for example, that's one of the muscles involved in maintaining your posture all day long. It doesn't require a lot of power to maintain your posture, but it does require a great deal of endurance. Those muscles are going to keep contracting until you lay down. And so if we look at your erector spinae, we find that they have a relatively high proportion of slow oxidative, SO fibers. So those are small in diameter, but they have lots of mitochondria, and they're able to produce ATP all day long. They're not easily fatigued. And the same thing would be true for your diaphragm. You need to keep breathing 24 hours, 7 days a week. Your diaphragm cannot fatigue, because if it stops contracting, you will go into what's known as respiratory arrest. And so likewise, we find many slow oxidative fibers in your diaphragm. Let's think about your arm muscles. Your arm muscles don't actually have to do all that much all day long. Rather, we rely on our arm muscles, generally speaking, for fast, powerful actions. And so our arm muscles can fatigue quickly, but they do need to be powerful. So we find many of those fast glycolytic fibers, the large diameter fiber types, in the muscles of the arm. And then our leg muscles have a mixture, more of an even mixture of fiber types, because we tend to walk around all day long. And so we do need those SO fibers for endurance, but sometimes our leg muscles need to give us these powerful bursts of movement, like jumping and kicking. And so we do have these fast oxidative glycolytic fibers. Those are those intermediate fiber types, and they're more powerful than the SO fibers, but they also fatigue more quickly. In this image, we can compare the three different fiber types with respect to their fatigability. So if we look at the vertical axis, we see tension in each one of these three panels. And if we look at the horizontal axis, they all represent time. And so we can look at the tension that is developed over time. And we see differences in the fatigability of these three muscle fiber types. Up top, we see the SO fibers. they can go forever and ever and ever. And I know that's an exaggeration. Can muscles really contract forever? No. But they can go all day long. So up top, the SO fibers are in muscles that have they're in higher proportion in muscles that have high endurance, like your postural muscles, like your diaphragm. And then down below at the very bottom, the fast glycolytic fibers, those are very powerful cells, but they also fatigue very quickly. So they're for short bursts of activity, and the muscles in your arm have a higher proportion of those FG fibers. And then we see the intermediate fibers in the middle there. They have a moderate level of fatigability and they have a moderate level of strength. This image comes out of your textbook. I love it. I feel like it very cleanly and clearly provides a lot of information. It's a very elegant image. And so we'll spend a bit of time talking about panel A and panel B, and they are represented. As you might see the connection immediately, they are connected by color. So let's look at panel A. In panel A, we're looking at a cross section through a particular muscle in your body. I don't know which one, pick your favorite muscle. I like the sternocleidomastoid. That's a muscle that runs sort of from behind your ear to your clavicle. And any given muscle is going to have a mixture of the 3 fiber types, which we see represented in panel A. We see the slow oxidative fibers represented in green, and look, there's a small diameter there. These are the weakest of the 3 muscle fibers because they are smallest in diameter. The second type of muscle fiber is represented in orange. Those are the intermediate fibers, the FOG fibers, and they are medium in diameter, so they're medium in strength, and they are also medium in their endurance. And then we have, in purple, we have the largest muscle diameter, the fast glycolytic fibers. Those are the most powerful cells because they're biggest in diameter, and they also fatigue very, very quickly. And so continuing to talk about panel A, we see that this muscle, the sternocleidomastoid, for example, includes a mixture of these 3 muscle fiber types, and each motor unit includes just a single muscle fiber type. So we can see motor unit number 1, according to the label, motor unit number 1 includes all of the slow oxidative fibers shown there. So how many muscle cells are in motor unit number 1? 4. We see 4 green muscle cells represented there. And then motor unit number 2 consists only of the fast oxidative glycolytic fibers. And so how many muscle cells are in motor unit number 2? 1, 2, 3, 4, 5, 6, right? There's 6 orange cells represented there. And then we have motor unit number 3, which includes only the fast glycolytic fibers. And so it'll take me a moment to count up the number of muscle cells that are in motor unit number 3, but let's go. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. 14 muscle cells are in motor unit number 3. Okay, so now that we have a handle on panel A, let's take a look at panel B. And on the vertical axis in panel B is whole muscle tension. So how much force is that sternocleidomastoid generating, that whole muscle tension? And then on the horizontal axis, we see time, and the little black arrows indicate the point in time when the next motor unit is activated, and that's called motor unit recruitment. So let's say I want to twist my head just a little bit, and motor unit number 1 gets recruited. So look at that, it's in green. So it's 1 motor unit number 1, and it's those 4 slow oxidative fibers start contracting. And look at how much tension is developed. Just a little bit of tension is developed. These are relatively weak muscle cells. And let's say I need to twist my head a little bit farther, or the sternocleidomastoid needs to generate more tension. We can recruit motor unit number 2. And notice that there's a little bit larger of a step up in muscle tension generated because these FOG fibers in orange, they have a wider diameter. There are more myofibrils inside each of those cells. And so when that motor unit is recruited, a bigger step up in tension is taken. And let's say I really need to turn my head or twist my head in a forceful way. Let's say I need to push against something with my head, and so that sternocleidomastoid needs to generate maximal tension. We can recruit motor unit number 3. And notice with the recruitment of motor unit number 3, we take this giant increase, this giant step increase in the amount of tension generated. This is because motor unit number 3 is the largest motor unit. It has the greatest number of cells, and all of those cells are the strongest type of cell in that muscle. The total tension generated by a muscle is influenced by motor unit recruitment, where each motor unit includes only one type of muscle cell, and each type varies in its ability to generate force. So if weak contraction is needed, if just weak contraction is enough to perform the task, to do whatever your body needs to do, only the slow oxidative motor units are recruited, and that generates a little bit of tension. If to accomplish that task, if we need to generate more force, then another type of motor unit is recruited. In this case, it's the intermediate fibers, or the FOG motor muscle fibers. And then if we need to generate maximal force, we will also recruit motor unit number 3 in this example. And motor unit number 3 is made up of the fast glycolytic fibers. So the most powerful cells come online to generate maximal force. So we do understand that there are differences, individual differences. People differ in their muscle composition. Some of us tend to have more FG muscle fibers and fewer SO fibers. And for other people, it's the opposite. Other people tend to have more SO fibers and fewer FG fibers. And we understand that this is partially genetically determined, but it can also be influenced by how you use your body, and that would be considered your environment. And so different types of physical activity or training can induce different changes in muscle tissue. Let's use these two types of physical activity to help us understand different muscle fiber types and the effects of training. So strength training versus endurance training. Think of strength training as weightlifting, and think of endurance training as running. They're two very different physical activities that can bring about different changes in muscle tissue. So strength training, so lifting weights every day, for example, will increase the diameter of muscle fibers because individual muscle cells will gain more myofibrils. So individual muscle cells get fatter. They increase their diameter because new myofibrils are built and added to the existing collection. And so these are mainly the fast glycolytic fibers that get fatter. And so what are the effects of weightlifting or strength training? It's muscle hypertrophy. That means that the muscle gets larger over time with repeated use. And we can contrast that to endurance training. Does running lead to particularly large muscles? No, not necessarily, but it does lead to your ability to keep running. And so endurance training leads to different changes in our muscle tissue. So with endurance training, you go for a run every day, and eventually you're able to run a 10K, and eventually you're able to run a marathon. What's happening in your body that allows you to eventually run a marathon or maybe even longer? Inside our muscle cells, we develop new mitochondria. Our mitochondria are very bacteria-like, and in fact, they used to be free-living bacteria before the origin of eukaryotic cells. And so your mitochondria can divide within your cells so they can multiply. What also happens during endurance training is there is a transition for some fast glycolytic fibers can become fast oxidative glycolytic fibers. So remember, the fast glycolytic fibers are the strongest fiber type, but they are also the first to fatigue. They don't have a lot of endurance. So for some of those fast glycolytic fibers, they can develop new mitochondria they can make more myoglobin. So they transition in their characteristics from an FG fiber to an FOG fiber. And then also a result of endurance training is angiogenesis, and that just means the growth of new blood vessels. So those muscle cells that now have a high demand for oxygen because you've started running every day, that high demand for oxygen, those muscle cells can send out growth factors that cause new blood vessels to grow toward them. And that process is called angiogenesis. And so it enables the cardiovascular system to supply a greater level of oxygen to those muscles. So elite athletes, we're thinking about Olympians. We are thinking about people who set the world record in their event. Is an Olympic athlete born or are they made? And the answer is both, right? So somebody is born with a particular talent or a particular fiber type ratio, as we are interested in today. And then, of course, the Olympic athlete trains every day for years. They dedicate their whole lives to being the best at their event. So elite athletes are the products of both genes and training. And we can compare two different types of elite athletes and think about their genes and their training and their fiber type ratio. So the comparison that we'll make is the marathoner versus the sprinter. These people are both runners. A marathoner is an endurance athlete, and a sprinter is a burst power athlete. So in the muscles of a marathoner, we find a much higher proportion of SO fibers relative to the other two muscle fiber types. And in the muscles of a sprinter, we find a higher proportion of FG fibers relative to the other two fiber types. And how do we know this? We don't need to wait for an athlete to die, because at that point they're probably, hopefully they're elderly and they've lived a nice long life, and it's been a long time since they completed an Olympic time trial, for example. But rather, we can take muscle cores from a living person. And so it's an instrument that bores into a muscle, like one of the muscles that makes up the quadriceps on the front of the thigh, and it pulls out a tissue sample. And we can examine that tissue sample and identify 3 different fiber types and look at their ratios. And so we have observed that marathoners have more SO fibers and sprinters have more FG fibers. And this is due to a combination of genetics and their training. So here are 2 athletes. These are both the top athletes in their event globally. Eliud Kipchoge is the world's best marathon runner. He holds the record for the fastest marathon time. And then Usain Bolt is the world's best sprinter. He holds the world's records for the best sprint time. And I'd like you to compare their body forms. Just look at their physiques. They're pretty different, right? One person is very muscular. The other person looks very slight, almost frail. Definitely not frail. But one person has a lot of muscle mass and the other person has not a lot of muscle mass. And so we can compare the muscle fiber type in both of these people and relate that to their respective events, burst power versus endurance. This is a fantastic video, definitely worth 2 minutes of your time, and I encourage you to stop this video and watch this sprinter versus marathoner video in YouTube. The link is also posted on Canvas in the recommended tutorial videos. You have probably experienced delayed onset muscle soreness. So this is not the muscle pain that you feel during the physical activity. So you're riding your bicycle up a steep hill and your quadriceps start burning. It's not that. Rather, this is the muscle pain that increases in intensity between 24 and 72 hours after that hard bike ride. And this is caused by local inflammation because during that hard bike ride, for example, you caused microscopic injuries to your muscle tissue and the connective tissue. There's a lot of connective tissue involved in muscle tissue. Every muscle cell is surrounded by connective tissue, and then your muscle cells are bundled into fascicles that are surrounded by connective tissue. And then, of course, each individual muscle is surrounded by connective tissue, and then muscle groups are grouped together by being packaged or surrounded by yet another layer of connective tissue. So during very vigorous physical activity, you cause these micro tears in the muscle tissue and the various layers of connective tissue. And these micro tears need to be repaired. And part of the repair process, this is a normal body function. There's nothing wrong with you here. Part of the repair process involves inflammation. And during inflammation, there is an increase in blood flow to a particular area. So in this case, it's a particular region of a damaged muscle. And those blood vessels also become more permeable. And so this allows for the faster delivery of oxygen and nutrients and substances that are required to heal those micro injuries. And it also results in the formation of signaling molecules that lead your central nervous system to perceive pain. And you can imagine why natural selection would have invented delayed onset muscle soreness, right? So if your quadriceps are really, really sore from that bike ride you took a day or two ago, are you equally likely to engage in that same hard bike ride again? No, because you're sore. It's your body's natural way of saying, hey, give it a rest. We need time to heal, and then you'll be good to go. You'll be even better after we heal. That soreness, those micro injuries, we now understand are part of training. They are integral to the process of our muscles getting stronger. And then the opposite of muscle hypertrophy, our muscles bulking up with repeated use, is muscle atrophy. So muscle atrophy is when our muscles decrease in size, and that's caused by individual muscle cells getting smaller. In response to a lack of stimulation or a lack of use, myofibrils get catabolized. They get chopped up. Those proteins get chopped up into amino acids, and then those amino acids get reused for other things. But as muscle cells decrease in the number of myofibrils they have, they decrease in diameter. So those muscle cells get weaker. And two of the most common causes of muscle atrophy are denervation atrophy and disuse atrophy. So denervation atrophy occurs when the motor neurons have been damaged. And so those motor neurons just can no longer deliver acetylcholine to those muscle cells. So those muscle cells never get stimulated. And so automatically those muscle cells are not used because the motor neurons are not functioning. This happened to a friend of mine that I went through K through 12 with. And in our senior year of high school, he was in a terrible motorcycle accident that severed some of the nerves in the brachial plexus. And so he lost the use of his left arm. And over the next several months in senior year, his arm was in a sling, and you could just see his arm getting smaller and smaller over time. So this was denervation atrophy, as those muscles were undergoing atrophy because those motor neurons were not able to stimulate them. And then there's disuse atrophy. I hope you have had the wonderful experience of watching your muscles undergo hypertrophy because you're using them more. And then you know the expression, use it or lose it. It's actually true. If you stop performing that regular exercise, if you stop lifting weights or you stop playing soccer or you stop cycling, you can watch those muscles lose mass. And that's called disuse atrophy. Or maybe part of your body has been immobilized in a cast. Maybe you broke your leg and you had to have your leg in a hard cast for say 6 or 8 weeks. And when that cast comes off, the muscles in that leg have all atrophied due to disuse. The good news is there, use it or lose it, well, you're going to use it again. So that muscle tissue quickly rebounds as new myofibrils are generated. Anabolic steroids. So when I put this word out in class, I ask students what comes to mind, and often the response is baseball, or sometimes it's a particular baseball player, or sometimes it's just sports in general. So anabolic steroids is a group of steroid hormones that are similar to testosterone, and they're called anabolic steroids. Well, they're steroids. That's a category of lipid. So they're definitely steroids, and they're called anabolic steroids is because they induce protein anabolism. They cause protein synthesis in muscle cells. And so they are abused by athletes. Abused is a strong word. They are used by athletes as performance enhancing drugs because they can increase muscle size and increase muscle strength by increasing muscle protein production, so myosin, actin, and the others. The dangers of exogenous anabolic steroids include the items listed there: kidney damage, liver cancer, heart disease, aggression. So when you see some of these professional athletes beat on their girlfriends or murder their girlfriends, I'm always suspicious of anabolic steroid abuse. And we'll talk more about anabolic steroids when we get to the reproductive system, right? Testosterone is an anabolic steroid, only in that context, it's endogenous. So something that arises within your body is called endogenous, and something that's delivered into your body from the outside is called exogenous. So in the context of athletics, we're talking about exogenous anabolic steroids. And then what's a muscle spasm? We've all experienced this, and sometimes they're sort of inconsequential or even funny. We've all had that little eye twitch, right? And we've all observed that in someone else. They have a tiny little twitch that we're just so tuned into facial expressions as humans. It's such an important part of the way that we can communicate. And then we have so many different facial muscles, and the motor units are so small, enabling us to have this intricate control over our facial expressions. That when a motor unit or 2 or 7 or a whole muscle spasms, it's very obvious in our faces. And it's pretty inconsequential, right? It's not necessarily painful. The person just looks funny, or maybe it's annoying. But then we can also experience painful muscle spasms, and that's often referred to as a cramp. So, maybe you're laying in bed at night and your calf muscle starts to cramp and it's causing you pain, and you leap out of bed and you start to walk around in order to get that muscle to stretch out. So in both cases, the facial twitch and the leg muscle cramp, those are muscle spasms. A cramp is a spasm that becomes painful. So a muscle spasm is an involuntary contraction of a muscle. And why they occur, we're still not sure. We think that dehydration plays a role. We think that electrolyte homeostasis plays a role. And we think that blood flow plays a role. The causes are not thoroughly understood, but for one reason or another, the motor neuron controlling that muscle, the motor neurons, I should say, one or more motor neurons serving that muscle are delivering action potentials. And those action potentials lead to muscle action potentials, which lead to muscle contraction. And when it's not voluntary, we call that a muscle spasm. All right, so this brings us to the end of, this is muscle lecture segment 3 of 3. So this is the end of our coverage of muscle, and what's up next is the nervous system. Thanks for watching.